SNAPP seminar. So welcome back to today's edition of SNAPP seminar. Let me start with a short bio of Philippe. Philippe is a research director at INRIA, and he teaches at Sorbonne University in Paris, where he got his PhD. His research interests include theoretical aspects of stochastic networks, random algorithms, and scaling methods of Markov processes. His current research focus is on stochastic models of molecular biology, and he's going to talk about some related work on stochastic models for neural synaptic plasticity. So without further ado, Philippe, the floor is yours.
Okay, thank you very much for the invitation. So today I will present you a work I did with a PhD student, Gaëtan Vignoud, for the last 3 years. And Gaëtan was also affiliated with Collège de France, where he's a co-advisor, is in charge of a biology lab in physiology. And so I will discuss the broad lines of what we did in 3 years. And basically, it was to understand in a mathematical way what plastic synapticity means. So this is what I will try to explain to you today. And so, as I said before, it's mainly about modeling. Of course, there are mathematics, but they will not be so present, because I think the important thing is to have what is represented by these models, and I will try to convince you that it has some relation with what usually think of synapticity. So, so I will have a quite long introduction, of course, mathematical models, and I will finish, if I have time, by discussing the mathematical results and what they say about plastic synapticity from this point of view. Okay, so in fact, we will not consider it very complicated, but this is the basic element of a neural network. It's a pair of neural network, neural cells. So you have what we call a presynaptic cell, and you have a postsynaptic cell. And the idea is that at some point, there is an action potential running on the axon, and which is connected in some way to the synapse of the other neural cells, and will send some electric signal to this cell. So this cell is what we call a postsynaptic cell. So, and this is what we'll study today, what mode of interaction of this. So when there is an action potential, we'll think of a presynaptic spike. And here, at some point, the postsynaptic cell will also fire some what we call the postsynaptic spike. And so the story is about what are the relations between these spikes, and what we say about the strength of this connection, what we call the synapse, between these two cells, and how does it evolve with time. Okay. So the one important thing is that it's not symmetrical. It's unilateral. So you have presynaptic cell. The information goes from the presynaptic cell to the postsynaptic cell. Okay, so you don't have kind of symmetry. So it's a unidirectional signal, and the presynaptic spike is issued from the presynaptic cell A, and the postsynaptic cell is with a cell B. Okay, and the synaptic weight, in fact, is the increment. So when the presynaptic cell sends an input to the postsynaptic cell, it increases, in fact, and this is interaction, it increases the membrane potential of the postsynaptic cell, and increase the membrane potential by some value W. This is a synaptic weight of the connection. So the larger W is, the stronger is the increment of the due to a presynaptic spike. Okay. Okay. So and this is our main quantity of interest here. And and so that it's a quantity which is which has a slow variation with time. Okay, I will come back later on the numbers related to this time scale. And in fact, it depends. So this is what the experiment shows on past synaptic activity. And the trouble is that it depends in a complicated way of the past synaptic activity. So, and this is where the modeling problem is, how we can represent this complicated functional of which is to express the increment of the synaptic weight with respect to time. Okay. And what we mean by past synaptic activity is just simply all the instant of previous, past, and post synaptic spike. Okay, so in the literature, this is referred to usually as STDP, which means spike timing dependent plasticity. And it's an empirical model which in fact says that my synaptic weight is dependent on the instant of the pre and post synaptic spike in the past. Okay, and so this is where we will what we will try to do carefully, how to describe that. Okay, so I will try to explain what is an experiment in this domain. So this is a classical experiment doing by a biologist and a biophysicist. What they do is just you take some real number, delta, and you what you do is you after a presynaptic spike has been emitted for minus delta milliseconds, you make the postsynaptic neuron spike. Okay, so this is what we call a pairing. You pair, so you impose that after a presynaptic spike, you have a postsynaptic spike. Okay, and you do that with some frequency, a certain number of pairings. Okay, and during a time you record the amplitude W, and W is just the jump of potential of the postsynaptic neuron. Okay, and and then you do that for many values of delta, and this is what you will get is you will get the STDP curve, and this is what biologists do. So I will not describe the experiments I have seen, but you take a slice of a brain and you connect two neural cells, and you send some input signal to make these events happen, and then you measure the jumps that you observe on the potential of the postsynaptic spike. And this is what and then we get an STDP curve and the function phi. Okay, and so a typical experiment. So this is where for a mathematician it's a little weird. So the experiment. So here the delta t, it means that you have a postsynaptic spike after a presynaptic spike, and you look at the increment of the weight, and you see in this case that the increment is positive. So all the small circles are the experiments, and here it is the contrary. You have a postsynaptic spike before the presynaptic spike, and you see all the circles tend to be negative. So the increment is negative. So, and this is what we call the Nebbian rule. And if you have a postsynaptic spike following a presynaptic spike, then you reinforce your synaptic weight, and otherwise you decrease it. Okay, so you have this continuous curve, which is, I would say, a kind of approximation of the circles. And this is something funny when you discover that for the first time. So this is the STDP curve, and usually it can be taken as exponential random variable. And this is just a kind of approximation of this experiment. Of course, the experiments are very noisy because you measure potential, and it's very precise. So this is why I guess the approximation is over. So this is in any paper in this literature, you will have this kind of curve, and this is well accepted, I would say, as a representation of STDP curve. Okay, I will mention, so there is a huge literature, and mainly it's biophysicists who are doing these kind of things. So the protocols, it started in 1997, these protocols of pairing post and presynaptic spike, and then introduction of STDP function, and then some proof that this is a biologically plausible representation of what's going on at the synapse of between two neurons. So I will come back to that later too.
Philippe?
Okay, of course, it's not something which is it's phenomenological model. Just it's a way of trying to represent the time evolution of synaptic weight by provoking this this pairing. And of course, it's not a biological phenomenon, a pure biological phenomenon as seen. So it's more a representation of the impact of synaptic activity, which is provoked in this case. And if you look at these models, so there are basically two main classes: pair-based models and calcium-based models. I will describe them. And with these two classes of models, you have basically all the models used by physicists and biophysicists to represent synaptic activity between two cells. Okay, so as I said, synaptic weight is the kind of functionals of some pairs, and this is where there is a modeling issue. There are some pairs of pre and postsynaptic spikes that have more influence than others in the synaptic weight. And the pair-based models are about that. So there are several models, I will present them. So you consider a pair of instant of pre and post-synaptic spike, so T pre and T post. Okay, so for every pair, you do that. And the variation of the synaptic weight, so this is a model, is the variation of the synaptic weight at the time, the max of these two quantities, can be expressed as a function. So this is my STDP curve of the difference T post minus T pre. Of course, if T post is greater than T pre, this is the increment at a post-synaptic spike, okay? And conversely, if T post is less than T pre, then this is a negative quantity. And you represent your increment like that, okay? And what we assume is that for distant points, they have no influence on each other. So if the two pairs are quite far away, the contribution is negligible. So here, I insist that this is the contribution of the synaptic weight due to this pair. And of course, you have as many pairs as you have pairs of pre and post synaptic spikes. Okay. And of course, the additional assumption is after some time, a given pair has less and less influence. So we have a leaking term, which is an exponentially decreasing term, which modulates the increment of a given pair. So as time goes by, then the influence of this pair is vanishing in the model. So this is the main assumption for this pair-based model. So I will describe, and so because I said some pairs, so and the different pair-based models are distinguished by the set of pairs which are chosen to describe this the time evolution of synaptic weight. So you have all two rules, and you take any pair which before time t. Okay, you have a nearest neighbor symmetric rules, and it works as follows. If you take a pair T pre and T post, then you consider all the pairs so that you don't have any postsynaptic spike between them. Okay, so if it's a postsynaptic spike, then you include this pair as long as you don't have any other postsynaptic spike before the last presynaptic spike, and similarly for the presynaptic spike. Okay, so you just localize the pair that you choose to be included in the function of your synaptic weight. And you can reduce it much more than that by considering the pairs T3 and T post so that you don't have any spike between them. Okay, and so this is represented in this drawing. So here on this line, I have the presynaptic spike. So I have one presynaptic spike here, and I have postsynaptic spike here. And so I will consider all these pairs like that in the all-to-all model. In the nearest neighbor model, I'll just consider all the pairs so that there is no postsynaptic spike, so only this one and only this one. This one is not counted because I have a postsynaptic spike here. And the nearest is even simpler. You just consider this pair, and this pair. Okay, so is it okay for the moment? There's any questions? Okay, so I continue.
Philippe, can you hear us?
Okay,
can you
so
hear us?
Fabian rule is, as I said, it just says that you put some positive increment if the postsynaptic spike is after presynaptic spike. Just means that if T post is greater than T3, then this part is positive, and otherwise it's negative. Okay, and so it has been shown in experiment that depending on the part of the brain, you have several populations of neurons. It holds for several of them. But the interesting fact is that anti-Hebbian rules also exist. So there are some parts of the brain where you have the contrary. And it leads to, by the way, to some interesting behavior which are not fully understood. Okay, so this is for what I would say for Hebbian rules. So here we have typically Hebbian rules, as I mentioned before. Okay, so how do we describe that? So if we think of the instant of pre and postsynaptic spike, then we represent our processes by point processes. So we will denote by n lambda the point processes of the point process of instant of presynaptic spike. And lambda stands for the intensity of the process, and of course we'll define the integral in the usual way. The sum of the function at the point of n lambda, and n beta x will refer to the postsynaptic spike. And so x, I will define beta is a function and x is a process. So, but for the moment, I will quickly describe it. But this is also a point process, which will be a little more complicated to describe the postsynaptic spike. So I have to. So for the moment, we just need these two point processes. And the synaptic weight, so if what I said with the words before, by describing my pair of pre and post-synaptic spike, in fact, just can be represented like that, that the synaptic weight at time t can be expressed as a functional of this kind of quantities. So you have a double integral with respect to the pre and post-synaptic spike, okay? And you have so v is post and u is pre, and so the increment at time v, let's say if like v is greater than u, this is phi 1 of v minus u, and at time t it is reduced by an exponential factor. And so this is for given u and v, this is the contribution of the pair uv at time t. And of course, if you want, you integrate over all uv between 0 and t, and you do the same thing for the post and pre pairs. And so we'll have functional direct. So very quickly, you have to handle this kind of functionals. Okay, so this was mentioned by physicist Morrison and others in 2007. And in fact, when we deal with this mathematical model, we just have to analyze these double integrals. And of course, the difficult part is the postsynaptic, which are not so easy to handle. Okay, I will come back to that. Okay, so this is for the all-to-all model, because I just integrate with for all the pairs of u of n lambda and beta x. Okay, so this is the all-to-all model. It can be shown that we have that. And if you look at the nearest neighbor symmetrical, then you just, for a given postsynaptic spike, you just look at the last presynaptic spike before V. And you look at, so V minus this quantity is just the time between the last presynaptic spike before V. And you, of course, the increment at the time is phi1, and at time t, it's with exponential decay. Okay, and so, and Tn of x is simply the first point of n of the point process n before x. Okay, so it's less pleasant than the old dual model, but still you can represent it in some way as a functional of the two point processes. Okay, so this is what I wanted to say about pair-based model. So the other class I mentioned are calcium-based models. Calcium-based models are in fact more, I would say, biologically oriented, but this is also an empirical model. It just say that you describe a quantity which we usually describe as the concentration of calcium. Calcium is playing an important role in the transmission of the electric signal from the pre and post synaptic cell, because you have in the membrane of the synapse ionic channels which let go in calcium ions, and calcium ions are believed to be one of the representative of the synaptic strength, although it's not really established on a biological basis. Okay, but these models are about that. So just say that the calcium, the concentration of calcium, so here we'll take the C of T, concentration of calcium, is increased by the quantity C1 when I have a presynaptic spike, phenomenological, and it is increased by the quantity C2 when I have a postsynaptic spike. So, and I have an exponential decay, gamma. Okay, of course, C1 and C2 can be negative or positive, depending, and depending on the model you choose. And in these models, so this is due to Grotner and Brunel, and they are quite popular, by the way, that you represent the synaptic weight as functional of a kind of occupation measure with respect to the level of calcium in your synapse. So if your threshold is greater than some quantity theta p, or for the p usually for potentiation, so meaning increasing your synaptic strength, so in particular the b sub p will be positive, then your synaptic weight will be a function of this integral. And if your concentration of calcium is above some threshold theta d, then you have a reward with quotes BD. And in fact, so we have a kind of functional of that, which represents the synaptic weight as just the functional of the calcium concentration at some given point. Okay? And of course, you have also this integration with respect to all the instant between 0 and t. So it's not an instantaneous increment, just an integral of a time interval. And this is, as I said, because of the synaptic weight, it's just slowly evolving. And this is one of the reasons you don't have instantaneous quantities, but more integrals over some time interval. Okay. Okay, mathematical models. Even though we already spoke of mathematics here, but it was more for modeling. So how do you represent the membrane potential of the postsynaptic cell? And usually, so this is a fairly classical way of representing. So x of t will be the membrane potential, the time evolution of the membrane potential of the postsynaptic weight. Okay, so you can see that for when you have a presynaptic spike, then the membrane potential jumps by W. And this is what exactly W is. This is a jump when you have a presynaptic weight, a spike, sorry. And n beta x of t is a postsynaptic spike. And when you have a postsynaptic spike, you have a negative increment of the membrane potential. So we'll take some example. So, and this term is what we call the leaking mechanism, that if nothing happens, the membrane potential will vanish or will go to some rest potential. Here, the rest potential is taken to 0, but it's not, this doesn't change anything of having another value, okay? Another entity is the instant of presynaptic spike, as I said. And so n beta x is in fact defined as follows. You have an activation function, beta of x. So beta is called activation function. So it means that when my membrane potential is at x, the rate at which the postsynaptic fires, meaning that it will have a postsynaptic spike, is beta x of beta x dt. So beta x is the rate at which it spikes, okay? And this is our process, okay? Okay, so let's take some example. If g of x is 0, so the postsynaptic spike do not have any influence of on the membrane potential. So we have a classical differential equation, which is a short noise process associated to the point process n lambda. So this is a classical process that which is used in many areas, as I guess as many of you know. And if you take g of x is x, it means that if, when you have a postsynaptic spike, then you jump of minus x of t, so x of t becomes 0. Okay, so when you have a postsynaptic spike, the resting potential is x of t is taken to its resting potential, which is 0. Okay, so and this is a very, very classical model of the neuroscience in the neuroscience literature, which is called the leaky integrate and fire model. So, and it is widely used. So basically, this will be the model that we'll use. And the assumptions, the probabilistic assumptions are simple. In our case, we assume that the postsynaptic spikes are Poisson processes with rate lambda, and the n beta x is a Poisson point process, of course, non-homogeneous Poisson point process with intensity function, which is beta x of t. Okay, and so, and this is where it's the things turned out to be difficult because x of t is itself the solution of stochastic differential equation, and you take n beta x, a Poisson process with an intensity function beta of x of t. Of course, there is no problem for the existence of such quantities, but it turns out when you try to do some mathematics on these objects, and this is where difficulties arise. Okay, so I'll just have a brief look at the mathematical literature. So mathematical literature is mostly devoted to one neuron, in the integrating fire model in particular. There are some works on population of neurons with PDEs, in fact, several of them, and few results on the population of neurons with mean field results. So I just mentioned quite a few, but now there are more and more. But there are basically, if you don't have a mean field result, the literature is quite scarce on population of neurons. And if we are looking at, of course, all these works are assume that you have a fixed synaptic weight. There is no question of time-varying synaptic weights, of course. And if you look at the time-varying synaptic weight, then you have a huge literature in physics, really huge, and for some time now, and very few mathematical models, and for some marginal models, I would say, of plastic synapticity. So this is quite an open domain from this point of view. Okay, so I will try to describe what we have been doing in this context. So of course, I will have to be a little more precise in the way what the functional of pre and post synaptic spike. And this is what I'm going to try to explain to you. For the time evolution of synaptic weight, we introduced two important processes, omega p of t and omega d of t. So basically, omega p of t is a process which will encode the memory of the past synaptic activity, which will lead to some potentiation. Okay, and similarly for depression meaning that you will decrease your synaptic weight. Okay, and so omega p and omega d will incorporate all the past activity, so that at some time t they will represent the potential of potentiation or depression. Okay, so it's done in this way, that the time evolution of your synaptic weight is a functional of these two quantities, omega p and omega d, and of course, of the current synaptic weight. And the assumption is that it's an increasing function of omega p and a decreasing function of omega d, okay, which is logical if you think of what potentiation and depression means. And an example, when you take threshold AD, you have this kind of things, but you can have also power of polynomials here and here. So this is one of the typical quantity which is used in some mathematical models of this time evolution. Okay, so and these quantities are complicates somewhat because they are not really related to x of t or w of t. And we have to describe them in more precisely, in fact. So for the moment, it's quite vague. So the way it's done is just by introducing a set of cellular processes, zi of t. So it's a vector. And in fact, the zi of t will play in some way the role of calcium variables. But you assume that you have L such variables which will represent as cellular processes. Okay, and these cellular processes will evolve according to the pre and post synaptic spike. Okay, according to this stochastic differential equation. Okay, so the zi of t can be thought as a concentration of some chemical component created by the pre and post synaptic spikes with some convenient coefficient. Okay, and okay, and our variables omega p and omega d can be represented in this way as a functional of this vector of the concentration of chemical components at some presynaptic spike, that's postsynaptic spike, and with some deterministic term. Okay, so this is the time evolution of our memory, in fact. If you solve that, then you have a kind of a set of differential equations, stochastic differential equation. So the first one is the membrane potential. The z is the vector of concentration of chemical components, and the omega are the memory. And finally, w is my synaptic weight. And the game is what can we say in such a context of the time evolution of the synaptic weight when t gets large? Now, this is the question that we'll look at. And of course, in the present state, not much. And this is where we will have to rely on scaling techniques to be a little more precise. Okay, it looks to be kind of artificial model, but in fact, we can prove, and this is what we have done that most of the pair-based models I've shown to you, and most of calcium-based models of the structure can be represented by this kind of formalism. And this is in fact something interesting, because it means that if you have some kind of result for this state process, then you will be able to have some information of the basic models of STDP in general. So these papers are referring to most of the models used, and in fact I would say 90% of them can be represented by such a framework. So I didn't tell you how the all-to-all model, for example, could be represented like that, but it's not difficult to see with some elementary stochastic calculus. So I will skip that. So as I said, there is little hope that you can say anything in such a state. So we have to introduce some scaling feature, and there are good reasons for that. Okay, so cellular processes. So it means that the processes that you look, which are at the molecular level in the synapse. So typically, spiking mechanism, the ions flowing through the axons, or the concentration of calcium. So you just count the number of ions in this within the cytoplasm. And usually, all these processes are in a time scale of several milliseconds. So typically, it's between 5 and 20 milliseconds. Okay, so very fast processes compared to the synaptic weight, where in the experiments I mentioned to you, usually the changes arise after several seconds, even minutes after one of the protocols I described to you of pairing protocols. Okay, so we have a different of time scales, and the idea is just to use that the fact that this process will evolve very quickly towards some kind of equilibrium, and we will be able to describe the time evolution of W with respect to this instantaneous equilibrium. Okay, so in other words, just try to have an averaging principle. Okay, so the cellular processes without anything are that, and the idea we are going to speed up all of them just by speed up by a factor over epsilon. So the Poisson process is accelerated by epsilon, so it iterates lambda over epsilon. The time also, of course, is by 1 over epsilon, and similarly for the postsynaptic spike. Okay, so you speed up the pre and postsynaptic spike, and you look at the x epsilon, z epsilon processes. So this is what we call the fast processes, the cellular processes. And for the slow process, so typically the memory, of course, you keep the presynaptic spike speeding up the presynaptic spike, but you moderate its influence by scaling factor epsilon, so that the change will be O of 1. Okay, so in this way, you have an O of 1 variable. Okay. So, and of course, we will be interested the behavior of these two quantities and this quantity when epsilon goes to 0. Okay, and so this is okay. So the first process is the x and z process when the synaptic weight is fixed to W. Okay, you can see here that W is fixed, and I call xW, ZW, the cellular process sees when W is fixed. Okay, so solution some stochastic differential equation, and quite simple theorem just tells you that there is an invariant probability measure pi W. It's done with standard technique of Lyapunov. It's annoying because you have a continuous state space, so you have to worry about some technical condition. But basically, this is a simple result. It just tells you for a fixed W, my XW and ZW have equilibrium pi W, and that's all. Okay, and so if we want to study the behavior when epsilon goes to 0, we have so this is classical when you deal with averaging principle. You just have to look the occupation measures associated to fast processes like this kind of quantity, integral of that. But in our case, we have also to deal with kind of occupation measure, but with respect to the postsynaptic weight. And this is one of the technical difficulties of this problem, is just to prove the tightness of these quantities. And it involves technical estimates associated to short noise processes. So this is one of the difficulties of the problem. And finally, to prove that these occupation measures converge to a quantity related to invariant measure of my fast process. Okay, the pi w of s. Here it will depend on time s, and this is a result which takes some time to prove by several arguments. It's several steps, in fact, but this is a key result. Okay, so these are technical results which are necessary to prove our main result, because now if you look at my memory from process, then you can represent it as an integral like that, and then you can see that we have all the ingredients we want, because the previous technical steps allows us to describe the limit of these all these quantities. And in fact, in particular, we can show that these processes are tight, and the result is averaging principle. There is some fixed quantity S0, which is deterministic, so that if I look at the process W epsilon, omega epsilon, then this is a tight process, but it is stopped at S0. So this is important. Okay. And any limiting point, it satisfies this kind of equation. So it's a kind of ODE, but with three functions, omega p, omega d, and omega p is related to W of s because of the dependence of pi ws, and W of t is related to omega p of t and omega d of t. Okay, so this is the main result that we have, and of course, it's not a convergence result, as you can see. It's not on the whole real line because it's for some fixed S0, and I would say this is the best we can do in the present state. Okay, so of course, when you take examples, you can do better than that, but this is really the basic result that we have. The tightness result is okay, and the convergence. So tightness result tells you that you have limiting points. Okay, so what you need after that is just to show that you have only one limiting point, and for that you need some kind of regularity properties for the invariant measure pi w, that it's locally Lipschitz. So you can do that in a case by case by using Lyapunov function. So regularity of invariant measure with respect to a parameter. So it can be done. In your setting, it's not so easy, but when you look at examples like we have done for pair-based model with all-to-all scheme, we can prove this kind of result. So in this case, when you have this property, you have convergence to a unique solution. The other point is S0. So this is an interesting quantity. It's kind of blow-up time, in fact. And what it says is that when this time is finite, it means that your synaptic weight is blowing up at 0. That is, in fact, if you think of the model we had before, it means that when you have a presynaptic spike, you have immediately a postsynaptic spike, because the strength of your potential is so that presynaptic spikes just cause quickly a postsynaptic spike. So this is a blow-up mechanism, and I will give you an example where you have this property. It's not a pathological, at least from the mathematical point of view, pathological result. And the dual phenomenon is when you have S0 is infinity and the limit of W of t is 0, that's it, the synaptic weight is vanishing. It just tells you the opposite, that the presynaptic spike has no influence at all on the postsynaptic cell. Okay, and of course, the interesting result is between them when you have convergence of a synaptic weight to some positive quantity W. Okay, so I will take a simple example by using one cellular process Z, where you have very well B1 when you have a presynaptic spike and B2 when you have a postsynaptic spike, and you just look at the time evolution of W as driven by the postsynaptic spike at the time of and with the weight of the Z at that time. Okay, and the activation function is a affine function nu plus beta 0x. So nu can be seen as kind of external input. Okay, and beta x is proportional to the membrane potential. So this is a simple example. And, and the asymptotic ODE is like that with a polynomial of degree 2. And you have two cases. If the discriminant of this polynomial is positive, then your S0 is infinity, and you can describe your W of t like that. And otherwise, W of t can be represented by this kind of function. Okay, so in this case also, you can have a blow-up mechanism. Okay, so, and remember, I will just, so this is an all-tool model. Okay, and I will, the all-tool model is all the pairs, and I remind you that the nearest symmetric neighbor is just the pair before, and nearest reduced is just without any spike between the two instants of the pair. Okay, so I will conclude with some mixture of experiments and simulations. Okay, so we can do some kind of calculations, as you have seen. This is the result we obtained. We did, in fact, other examples. And so if you look at the all-tool model, and so here, I have to remind you the model. So B1 is the weight when you have a presynaptic spike for the increment of the synaptic weight, and B2 is the weight of the when you have a postsynaptic spike. And the drawing I have here, B1 is on this axis, and B2 is on this axis. Okay, so you can see that with B1 and B2 positive, we have a red zone, meaning that if you start from this zone, then your synaptic weight is going to infinity. Okay, and if you start from this region, like the B here, the green one, then you converge to some fixed point. Yeah, and if you, and there is also this region where it's an equilibrium point, but which is not stable. If you start above, then you diverge to infinity, and if you start below, you just go to zero. It vanishes. And here, if you start from the blue zone, then you're converging to zero. So this is what we have in the all-tool mechanism with an external input of rate 1. So if we look at the another scheduling, so linear stable, you can see that you increase the stable region. The green one, for example, and the orange one is increased somewhat, just because, and this is the dependence of the mechanism when u is equal to 1. Okay. And of course, I should say that some part of this drawing, we don't know how to, for example, this part, it's only simulations. We don't have analytical formulas for these parts due to this activation function. So it's only partially analytical, I would.
Oops, it looks like we lost Philippe.
Oops, he doesn't know that we lost him, I think.
Doesn't know?
Can you unmute him? Can you unmute him yourself?
Yeah, I should make him a co-host. Philippe, we can't hear you.
Oh, okay.
Okay, now we're good. Now we're good.
Yeah, we're good?
Yes.
Okay, sorry. But I am almost done. So if you look at these two, so we have this green zone, which is quite important. And if we look at the reduced one, it's very thin. Of course, simulations are not that coarse, but are quite coarse, but and we have kind of strange phenomenon. And however, it seems that either we go to infinity and we go to zero, but the zone between them seems to be quite small. Okay, I will conclude to say that, of course, once you have this averaging principle, you have a kind of ODE, which is not that easy to understand. And more important, that stability properties, that is the asymptotic behavior of synaptic weight, has to be investigated. And of course, it's I would say case to case, depending on the model study. And there are some models where we can do such analysis, but for others, it's a little more complicated. Also, because of the invariant measure pi w, which is not known. And this is one of the main problems. You don't know sometimes the exact invariant measure of the fast processes. And this is a classical, I would say, problem of arranging principle. There is another potential problem. It's just the STDP function phi has been taken exponential, which is good, but sometimes it's not a good formulation. But in this case, we are living in some way the Markovian setting, and so averaging principle is a much more difficult problem to formulate, even in this case when you don't have some nice Markovian properties. And last but not least, and this is what we're currently doing with Gaëtan, is just by considering population of excitatory and inhibitory neural cells. And in fact, this is related to problem of related to Hawkes processes. Okay, and we did some papers, as you can see, just to present the models and the proof of the result. And that's all.
Let's thank Philippe. The floor is now open for questions, so feel free to unmute yourself and ask questions.
Maybe I'll ask a question. Oh, you go, Takis, sorry.
No, my question is very generic. It's all right, I'll go.
Mine's pretty generic as well. You go. Yeah.
Okay. What are I mean, because you focused on the models and so on, what are the things that somebody in the area, I mean, not an applied mathematician or probabilist, would like to know? I mean, what is it that they care about? I mean, I don't quite get it.
Well, can you repeat your question?
No, I know. I just want to say, what is it that somebody, a neurosurgeon or whatever in this area, would like to know from these mathematical models? I mean, communication network. Somebody might want to know the delay of a packet from. I don't quite know.
No. We are living in a more complicated world.
I'm sure.
It's a very complicated phenomenon. And even the physicists are describing them with, I would say, rough. Basically, I would say, at least in my views, the way I'm doing this work, we would like to understand the qualitative properties of the experiments. Just can we have some mathematical models where the qualitative properties of experiments can be represented? And this is what we are trying to do. And of course, my student used some data with STDP curves, but you have to be modest in this case because we don't, it's not like in communication networks when you can measure the delay and predict it and so on. We just want to understand very simple things like what is the behavior of the synaptic weight when T is large. Can we describe that with a convenient model so that the experiments can be explained in some way? Mm-hmm. Okay, and it's this is the way I look at it. And if you think of the pair-based model or calcium-based model, it's just to put some kind of formal framework to represent this complicated phenomenon of memory in general. And this is what it is about. And can we represent that just by using the instant of presynaptic spikes and postsynaptic spikes? Probably not. You have, I didn't mention to you that you have more complicated model where you don't take into account only presynaptic spike and pairs. You can use triplets because there are some phenomena which can be explained only with triplets and these kind of things. And I would say this domain is studying some very complicated phenomenon. And of course, we use some degraded model in some way. But with that, we hope to understand some phenomenon that we can observe in experiments. Like the typical experiment, just when you do this pairing mechanism, you do 50 and 100 pairing, and you see the time evolution of your synaptic weight. Can we have a model where you can describe these kind of things? For the moment, it's not that convincing. Okay. And this is really that. It's not a question of measuring, predicting the potential of the membrane neuron. This is why I said it's a more qualitative test, just if we can reproduce some qualitative phenomena observed in some experiment, then we'll be already glad of that.
And could that eventually may be used in order to design experiments? I mean, real experiments, or?
This is I did that in another domain in biology, just to design some other experiment. But I must say that in our case, we are already with existing experiments, and we try to figure out how we could describe them mathematically. So we are not at the level of proposing new experiments.
No, I understand.
I can tell you that the physicists have a lot of imagination for that, and there are many. So I mentioned the triplets, but you have many other STDP protocols that are used, and I don't think we are at the level of proposing new for some good reason. We don't have that. We are struggling already to understand this pairing mechanism, how they influence the synaptic weight, and it's already not that easy, at least in my view.
Okay, all right. Thank you, Philippe.
Yeah, hi. I was just going to ask a bit about, I was trying to think about some of the processes that goes along. I mean, for the faster processes, can you characterize like their stationary distributions? So when W is fixed, so it almost look like, you know, kind of jumps up as a Poisson process, goes down exponentially, and then jumps to zero. Trying to think, is it, you know, is this quasi-reversible or something like that? You have things like with chemical reaction networks, you have these kind of timescale separations, but also reversibility. But there's, I'm guessing this W process you get at the end, you don't want that to be otherwise, because it wouldn't be very interesting.
And W is hopefully deterministic in the end.
But for the fast timescale things, is there anything you can say there about the stationary behavior?
No, the trouble is that we are not able, if we have a quite complicated cellular process Z, we are not that able to have an explicit representation of the invariant measure of the fast process. And of course, when the fast process is basically a two-dimensional process, X and Z, then we are able to do that. This is why we did some calculation I've shown you at the end, partially. Otherwise, I don't think we can have some more precise if we have more cellular processes. And this is one of the difficulty of this problem, in fact. And this is why but if our simple model already is not that bad. We can describe, in fact, the difficulty is also from the activation function. The activation function, you assume in some way that your postsynaptic cell cannot spike when its potential is negative. Okay, so you're obliged to have a kind of threshold at 0 for the activation function. And analytically, it's annoying because you have to, and this is where the trouble arises for the invariant measure when you write the invariant measure. Function like I mentioned, nu plus beta x positive part is very bad when you try to do some analytical calculation of the invariant measure, because you have discontinuity at 0. Okay. And this is so this is why we did this these examples with but there is a promising activation function with an exponential where you don't care, the exponential is always positive, and I guess we could do some more calculation in this case by assuming, in fact, exponential activation function means that once your potential is a little large, then you spike right away. Okay, and this is some way this is not a bad model for synaptic activity, in fact. But Still, as soon as you have averaging principle, you have this problem of expressing the invariant distribution, of course. And sometimes you can, sometimes you cannot.
Yeah. I mean, in principle, because you're going to speed up time anyway, you could change the model for that part of the system as well. I mean, I think you're like, why does it have to be continuous state system? You could have a discrete state system there instead. And when you speed up time, you
sorry,
might.
I didn't follow the last.
I was just saying, like, that, if you're going to speed up time and take the average with respect to the kind of X process, you don't. It doesn't have to be a continuous state space model there.
Oh yeah.
I mean, from a modeling perspective.
Okay, so I didn't mention that in my talk. Of course, you're perfectly right. Very good. In fact, we did some discrete model, the calcium-based model, with a discrete state space, and in this case, we have explicit representation of our invariant measure. Of course, because we take advantage that basically there are complicated functionals, but functionals of Poisson processes in a 2- or 3-dimensional state space, and you can represent most of the invariant measure with that. And in this case, of course, in the discrete case, we can do that. And in fact, it's not that bad, because if you think of calcium concentration, you don't speak of concentration, you just think of the number of ions of calcium. Then, so you have to represent your synaptic weight as an integer or so. And if you represent also the membrane potential like that, then you can have a perfect model, which is very close to the continuous model, but for which all these fast processes can be, you can express the invariant distribution of these fast processes, in fact. So this is what we did in one section of our papers, just to have a discrete version of that. And you're perfectly right. In this case, you don't worry about that, of course. And if you want to have, of course, you have to use a numerical procedure, but you have everything you want to describe your synaptic weight.
Yeah. Okay. Great. Thanks.
There are no questions. Let's thank Philippe again and end the seminar.
Thank you.